morning, ladies and gentlemen, both in presence and from remote, and welcome to this speech titled Technology Transfer and Collaboration with Private and Public Sector. My name is Davide Montesarchio, and I will moderate this talk on behalf of Fondazione Internazionale Trieste. It is an honor for me to moderate this speech by Professor Ian Matai. Professor Matai is a distinguished scientist, a member of several learned societies, such as the Royal Society in London and Edinburgh, and the German Academy of Sciences, Leopoldina. Professor Matai has made important contribution to our knowledge concerning cell biology, molecular biology, and biochemistry. He has been director of the European Molecular Biology Laboratory for 13 years till 2018, and since early 2019, he is founding director of the Fondazione Human Technopole. The topic he will be talking about has been broadly discussed, at least here in Italy, in the recent past, both in the academia, but also in the political debate. So I leave him the floor, and we'll meet later for the question and answer session. Thank you.
Thank you very much, Davide, and welcome to everyone. I'm sorry that I cannot be in Trieste, but I'm very happy that we have the possibility to connect via a remote connection to talk about what I will present, which are two topics. The major topic of the presentation is how academic organizations using Human Technopole as an example should engage in technology transfer and should work together with industry. This is a very important topic, and I'll come back to it. But I was also asked to provide a brief introduction to how the Human Technopole project, which is a new project which many people are interested in, is developing. So I will start with a brief introduction to the status of Human Technopole. As many of you are aware, the Human Technopole is located on the Expo Milan site, and it is located, the headquarters building is the Palazzo Italia of the Expo. The mission of Human Technopole is to promote and contribute to the improvement of human health and well-being. So it's a very broadly based mission. The decision that we've made is to focus our efforts on personalized and preventive medicine in research. So what I will tell you about is a program to focus around this area. We also intend to set up and operate scientific infrastructures, facilities, and services that can be made available to external users. We are beginning this, and I will talk about a couple of the facilities that we are starting with. We want to help drive innovation and progress by promoting technology transfer and by working with industry. Why is this important? Research is almost universally publicly funded. And the public has also a right to benefit from the products of research, from the discoveries of research. And because of the process of developing research discoveries towards either medical interventions or commodities that can be commercially available, it's essential to work together with industry because academic organizations, universities, do not have the capacity to take products, particularly products in the life science, all the way to market. And finally, the other mission of Human Technopole will be to disseminate scientific activities. In other words, training events for scientists, but also public events in which we try to explain new developments in research at Human Technopole and elsewhere to the public in order to make the public aware and to have a dialogue about the public, with the public, about where science is going. So we have been in existence for a bit more than a year. The initial research aim, of course, is to recruit people who will work at Human Technopole. We have been engaged over the last year in a number of recruitments, and in my opinion, many of these have been extremely successful. We've been able to recruit internationally. Only one of the people that we have recruited comes from inside Italy. Others are Italians who are returning to Italy, and others are nationals of other countries who are coming to work in Italy for the first time. We have decided to focus on 5 areas initially: genomics, both functional genomics and genetics and genomics, computational science, neurogenomics, and structural biology. The final area is a project which we're carrying out in collaboration with the Politecnico di Milano, which is called the Center for Analysis, Decisions, and Society. And in this center, we hope to use the methods of big data analysis to obtain information to help define strategy and to help the evaluation of the overall health system nationally. So in the genomics, neurogenomics, and structural biology area, we have already appointed heads of these research centers. In computational biology, we do not yet have a head, but we have 2 group leaders, and also we have other group leaders in the other 3. At the moment, we are just finalizing the recruitment of the head of computational biology and the head of the Center for Analysis, Decisions, and Society. In addition, we are in the process of recruiting more group leaders into all of these research centers. And we've made, for example, offers to 3 people to join the Research Center for Structural Biology in the recent weeks. We are having a high level of acceptance of our offers. I think people are very interested by the project and by the prospect of being able to work in Italy. And the people who are applying are a mix of Italians who are working abroad currently and people from other countries who are interested to work in Italy. And our new recruits are also a mix of these 2 groups of people. Our intention is to be an open institute that, as I said, will provide access to user facilities to external scientists. Those will include an electron microscopy facility for carrying out cryo-electron microscopy and for EM tomography. We want to have a visitor program so that people can come to Human Technopole to collaborate, to learn new techniques, to make use of equipment which they don't have in their own institutions. And we would like to develop bioinformatic data resources. I think one of the issues about current life science research is that it produces very large amounts of data. It's no longer possible to publish this data in the form of journal articles in its entirety. And so data resources which collect the initial data which is produced become more and more important, as do tools to extract information for that data. And we'd like to be active in this area. Finally, as I said, we will be engaged in training both internally obviously the people who come to Human Technopole as PhD students, as postdoctoral fellows, but also as starting group leaders, as starting principal investigators, will be provided with training. But also we'd like to have advanced scientific training of different sorts for external scientists, and we're planning that. And in relation to this talk, which is focusing on innovation and technology transfer, you may be interested to know that our plan for the first of these training events is actually not to have a scientific course, but to have a course on entrepreneurship for starting PIs, for starting group leaders from across Italy. It's a program which is aimed at enabling these people to develop an entrepreneurial mindset, to enable them to see, are there commercial opportunities in the work they're doing? And if there are, and if they're interested to develop those, how do they go about making the interactions with technology transfer professionals and also with funders in order to take these projects forward? And this first course, which will be a repeating course, the first of those, we hope to be able to hold towards the beginning of next year. It was originally planned for this year, but of course, coronavirus has delayed the implementation of these ideas. So I want to stop this, or to end this section by giving you an idea of how long and it will be before the Human Technopole infrastructure is completed. So we have we have Palazzo Italia, which is essentially completed. There are small things still to do. We have part of a data center that is a modular data center. And so we already have storage facilities and we are about to install high performance computing. The North Pavilion and the South Pavilion, which were previously expo pavilions, are being renovated in order to be turned into laboratory buildings. Palazzo Italia is not a laboratory building. And finally, incubator labs are being constructed. And actually, if you look at the Human Technopole Twitter feed, you can see how quickly these incubator labs are being constructed around what was the Tree of Life during the expo. And finally, we have planned a new building which will hold about 1,000 people, which will be completed roughly by 2025. I should say that the plan is at steady state that Human Technopole would be an institute of about 1,500 people with perhaps 1,000 scientists working there. So it will be a large-scale initiative as big as the biggest individual research centers in any country in Europe. And the Technopole site, the Expo site, is being developed as an innovation campus, the Milano Innovation District. The three initial tenants will be a research hospital, the Galeazzi Hospital, us, Human Technopole, and the science faculties of the University of Milano, who are again in the process of completing design projects in order to construct buildings which will enable them to begin moving to the site in 2024. Furthermore, there are ambitious plans and already some concrete moves to bring innovative industry onto the site, hopefully to work together with these medical and academically related institutes. So, as I said, we want to be an open institution. We want to have very good contacts with the national and international communities in research. That we want to have very good contacts with the health system, for example, the IRCs, but also other parts of the health system. And for the subject of the rest of my talk, we want to have collaborations and interactions with industry. So why is it important to build strong relations with industry, and how do you go about this? I've already explained that I think it's essential that research institutes demonstrate that they are the innovators which enable new industries to develop. And so I think it's very important that research institutes engage in technology transfer and engage with industry to demonstrate the value of basic research for innovation. This is a fundamental requirement if researchers wish to continue to be publicly funded. That's the rationale. What is needed? We need technology transfer professionals in each research institute that understand both the researchers and what they're doing within the institute, so they can help the researchers to identify what might be a useful discovery or a useful development or a useful technology, a useful machine which they have developed that could have a broader applicability in innovation. They also need to have a very good understanding of the field of biotech, pharma, and IT industries and what these specific industries are interested in. Because although it's the researchers, hopefully with an innovation and entrepreneurship mindset, who see potentials for innovation and discuss with the technology transfer professionals, it will also in Human Technopole be, the researchers will be free not to protect discoveries that they make. That will be their decision. But if they decide that the innovation can be protected and they are happy with this, they would work with the technology transfer professionals. The technology transfer professionals would generate intellectual property. The intellectual property would belong to Human Technopole. The researchers who were the inventors would benefit from any financial income that comes, but also the Institute, Human Technopole, would benefit. And I think it's extremely important in an organization to have a clear and transparent process so that everyone knows what they can do, what they can't do, what they can expect and what they cannot expect if they engage in technology transfer and in innovation and in the generation of intellectual property. But having relations with industry goes way beyond simply having a technology transfer operation that can be set up as a company or can be part of the initial organization. It also involves interacting with industry on many different levels. One of the ways in my past history that this has been very successful is in the development of equipment. Obviously, if you are running a facility with state-of-the-art equipment and you're in a research environment, you're constantly exposed to researchers who want to have the equipment be able to do something additional. One of the ways to allow this to happen, if it's of applicability more broadly, is to work with industry in order to add this capability to existing equipment. And this is something that we will do. We are already planning to do this in some areas, for example, in the automation of the generation of organoids and transformation of induced pluripotent stem cells, but also in imaging. A more ambitious type of interaction and a bigger type of interaction are public-private partnerships. This is where research institutes other public institutes like hospitals, for example, work together with industry to carry out projects. And I'm going to give an example, a set of examples which come from one of the people we have recruited, Francesco Iorio, who we have recruited to the Computational Biology Center, who has been working in a large-scale public-private partnership in the UK. And I'm going to tell you a bit in more detail about what he's been doing. So he's been working in an organization called Open Targets. Open Targets consists of a group of pharmaceutical companies, GlaxoSmithKline, Takeda, Bristol Myers Squibb, and Sanofi, together with the European Molecular Biology Laboratory and the Wellcome Sanger Institute. And the goal is to carry out research to help make the use of functional genomics and bioinformatic methods design better targets for drugs and point out, prior to drug development, potential drawbacks in specific drug targets. The reason this is necessary is probably all of you know that roughly 90% of drug trials fail before the drug gets even to stage 3, and roughly 19 of testing in patients, and roughly 99% of all drug candidates fail before they can be developed as medicines. The reasons for this are complicated, but both the companies and Wellcome and Embo were of the opinion that making use of genomic technologies and bioinformatics was one way to improve the selection of targets. So Francesco has been involved in various projects. This is one where essentially a group of 19 types of cancer, actually it's something like 1,000 different cancer cell lines of those 19 types, were systematically mutated by CRISPR-Cas9 screens, removing one gene at a time from each of those lines, to determine first which genes are required for core fitness, in other words, for cells to survive. But then the more interesting set of genes are the genes that are required not in all cancer types and not in all cell lines of each cancer type, but in a context that's specific. In other words, this is, if you like, in vitro personalized medicine. You're looking for the things which affect a specific type of cancer and a specific type of cancer cell line. Just as in personalized medicine, you want to find something that will help the specific patient you're treating if the general drugs do not help. So using data which has been produced by other projects, it was possible to make scores for a very large number of targets in, for example, colorectal carcinoma and ovarian carcinoma. And this was used to develop a software called Project Score, which is a publicly available resource which enables people to check how good a target, any given target is. One gene which came out of this screen in a subset, with a subset of the partners of Open Targets, was that it was discovered that in cancer types that have microsatellite instability, Werner syndrome helicase is a selective target. In other words, by removing Werner syndrome helicase from microsatellite unstable cell lines from colorectal carcinoma, or ovarian carcinoma, you reduce greatly the fitness of these cell lines. In other words, Werner syndrome helicase is not a target in all types of cancer, but only in a specific subset that suffer from microsatellite instability. And on the right of this slide is simply a confirmation that the removal of Werner syndrome helicase from these types of cell line by induced expression, by induced repression of the helicase greatly reduces the number of transformed cells in a tumor. And this is an experiment done in mice where the tumor volume is reduced after the treatment with doxycycline to turn off Werner helicase. The initial experimental demonstration relied on in tissue culture experiments in which you could see that when microsatellite instability was present, but not when it was absent, removing the Werner helicase greatly reduced the number of transformed foci produced from various different cell lines. Sorry. So these type of results are interesting both from the point of view of general knowledge about cancer, but they're particularly interesting for the companies in open targets because obviously they know, following such studies, which types of patient might respond to particular drugs. In other words, it's information in the field of cancer which leads to a more personalized treatment. This is a second example, which is really a completely informatic analysis, which was done by looking at mutations, copy number variation, DNA methylation, etc., in primary tumors from a large number of patients, and using genomic features to classify these patients into different categories that had similar genomic features. That information can then be used to propose what might be good drugs, existing drugs, to target these patient groups, or what might be good targets within these patient groups to try to develop drugs against. And so Francesco has been engaged in this project. It's a project which goes in circles, if you like, because the signatures are defined by the patient genomes. Cell line mapping is used to look for cell lines which represent tumors which don't have good in vitro models. These are generated into organoid type models and then tested in a high throughput way for their responses to drugs in order to make suggestions which can then be followed up together with the companies about what kind of drug might be described to these different patient groups. And finally, because we have a focus on structural biology and because we're working on health and well-being, I often get the question when I give presentations about human technopole, why would we choose structural biology? I think there are two reasons for this choice. I think one is related to the possibility it offers us to provide a new technology to the national and international community. By this, I mean the cryo-EM facility. But also, it's been my experience that structural biology is often crucial in the development of drugs. And I'm going to give you one example from my past. This is an example that was based on the work of Stephen Cusack at the MBL and his colleagues over many years. Stephen is interested in viruses and in viral replication. And for many years, he attempted to determine the structure of the influenza virus polymerase. Influenza virus, actually, like SARS-CoV-2, has a very interesting polymerase, which has specific features. So the polymerase, for example, has a module which removes the cap structure from cellular messenger RNAs and uses the cap structure as a primer to synthesize viral RNAs. So it both destroys cellular mRNAs and generates viral RNAs at the same time. There is no such activity in cells. And so for that reason, and because this is an RNA-based RNA polymerase, the influenza virus polymerase is potentially a very good drug target. But no drugs were developed specifically against it, partly for the reason that it seemed to be impossible to obtain the structure, the atomic structure of the polymerase, until Stephen and his colleagues worked with new methods for producing proteins, new methods for producing structures from very small crystals, etc., to obtain the structure of the influenza polymerase. They worked then together with a small molecule screening facility, an academic one, to identify lead compounds that inhibited the polymerase, and then set up a startup company, Savira Pharmaceuticals, to follow this up and to develop the lead compounds towards the market. So these developments proved to be very successful, and initially in collaboration with Roche, and later in collaboration with Takeda, a very effective flu drug was developed against the influenza polymerase, which is now marketed in the US and Japan under the name of Xofluza. It has the great advantage that it's the first drug that can be taken against influenza virus after the infection has started. So it inhibits the infection even when the patient is ill. It doesn't have to be taken before the patient is ill. So this is an example of the applications which can come from structural biology and why we've chosen to have this technology in human Technopole. So thank you very much. I hope I haven't gone over my time. I actually was supposed to see a timer and unfortunately could not see the timer.
You were right
so
on
Davide,
time. Yeah,
good.
you were right on time. Thank you, Ian. Thank you very much for your highlighting, enlightening speech. And I just remind to all the audience, both here present physically at Euroscience in Trieste and both remote from the internet, that now we have a question and answer session. You can ask your questions. For the people who are present here, just raise your hands. There is a microphone over there, and you can just have your questions. And for whoever is connected from remote, on your right-hand side, you should see the icon for asking question. Then I will receive them here and I can ask them to Ian. Please go on. We have a first question from the audience here in Trieste.
Okay, can you hear me?
Yes.
Yeah, okay. Good morning, my name is Marco Masia, I work as innovation consultant. First of all, congratulations for the project, it looks very ambitious, but I was really surprised that in all this ambitious project, the innovation part, at least the way you explained, is only with technology transfer and having like innovation brokers, meaning experts in science and also in business to bring up the innovation and nothing else than that. So do you plan to do something else, like building on the innovation ecosystem, having entrepreneurs in residence, contacts with investors, and things like that?
Yes, the idea is to do that. Obviously, the technology transfer professionals should have contacts with and should be familiar with investors and what they're interested in. I didn't speak about that when I was talking about the campus, but the reason we will not have space inside the building for spin-outs to start is that the main campus has a lot of space, which is intended for spin-outs. And so the intention is to make use of the main campus to obtain space. And when I talked about collaboration with industry, yes, the idea would be also to have industrial visitors, visitors from industry at Human Technopole. So these things are planned. I didn't go in detail about each of them.
Okay, thank you.
Yes, please go on. Another question from the audience here.
Hello, thank you so much for your very, sorry. You can use the other microphone. Okay, can you hear me?
Yes.
So thank you for the very beautiful and inspiring presentation. You mentioned that structural biology, of course, is crucial to drug design and development, but I think there is a general agreement that physiological factors also have an effect on the performance of drugs. And if I got it correctly, your computational, I'm a computational scientist, your computational
Yes.
group is, for the moment at least, strictly focused on structural biology. So the question is...
No, that's a misunderstanding.
Okay.
So
sorry.
Francesco Iorio, whose work on cancer I talked about, is one of the computational scientists. There is another who works on image analysis, and we are in the process of hiring a head of computational biology who has a different discipline. So there is no focus on structural biology in the
And what about
computation.
how would I say, the transport phenomena inside the body, the fluid part?
So this, at present, we have not yet hired people who are physiologists. A neurophysiologist, yes. One of the people who we're hiring in the neurosciences is a neurophysiologist. But that is something that either we will have to do in collaboration if we don't find someone that we can hire, or we will have to hire in the future. Remember, I've shown you that we're at the very beginning. So we currently have a small number of research teams actually, who are not mainly on site because the experimental labs are not yet ready to be used. But of course, we'll be building up substantially in the future.
Okay. Thank you. Thank you so much.
We have, yes, another questions.
Hello. Thank you for the very interesting talk. I have two questions, actually. The first one is about the course that you're gonna organize about entrepreneurship. I think it's a wonderful initiative, but why are you only offering it to PIs and not to younger researchers?
Simply for the question of numbers. So this is a course that will have a maximum capacity of something like 25 people. And so we thought that for the first course, we would concentrate on PIs. And in future, we will see, we may broaden the scope of the course. But actually, one of the worries I have and had about that course is that we will be overwhelmed by applications, because I know that a lot of people are interested. And so we built in to the first call the fact that we would offer this to PIs just in order to try to cope with the numbers. Otherwise, the selection would be extremely difficult.
Okay, thank you. And then the other question is about when you mentioned that you're going to also organize outreach activities to kind of disseminate the science that is produced at the Human Technopole. I was wondering if you already have any ideas on how to implement that part, or...
So for the moment, we've been doing that in a reasonably conventional way. So I, or the president of Human Technopole, or the scientists from Human Technopole, have been engaged in local events thus far. So events like Wired Italy, which is an innovation congress, which is run by the Wired magazine. We've engaged with the Science Night, the European Science Night. We've been participants in that. We've engaged with the STEM Careers for Women initiative and given talks at that initiative. So we've been engaging in a variety of ways with other organizations, including the Feltrinelli Foundation, for example, in engaging and talking with and having a dialogue with the public in various situations. At this point, it's still too early for us to say exactly which events we will organize in the future, but we are planning, for example, to also bring school children onto the site to let them see laboratories, to let them meet scientists. One of my experiences in science in the past has been that when school children come to a research institute, they often expect to find some kind of crazy monsters who are the scientists. And instead, they become very easily fascinated by seeing what scientists are doing, the type of equipment they use, using microscopes to look at pond water, very simple things, I think really fascinates children. And so one of the things that we will do is to bring school children also onto the site.
Okay, thank you. That's a very nice initiative.
I also have a couple of questions for you. We have a few minutes for the end. So the first one is that how much important is, in your opinion, the context around a research center or research institution for technology transfer? I'm thinking about Silicon Valley or the Milan Innovation District.
Yeah, so I think what is clear from just looking around the world is that areas where there is really top quality research. Silicon Valley was a particular area which was based around electronics and materials science. Boston, so the Cambridge, Boston, with Biogen, with MIT, with Harvard, with Genentech, is the world center of biotech, which was generated by the research institutions. Silicon Valley mainly grew out of Stanford University. In Europe, there are large science parks around the academic institutions in Munich, in Germany, in Heidelberg, in Germany, in Cambridge and Oxford, in the UK. These are the centers of innovation. And I think what you see, Milan is a center of innovation. Milan is, I think, the center for particularly for biotech, but also for IT in Italy, in fact, in all of Southern Europe. And again, I think this grows out of having excellent research institutes. So I think it's very important to have the environment. But the most important thing isn't that it has to be local. The most important thing is that the research institutes have the mindset that this is part of their function.
And talking about the mindset, like your initiative and many other initiatives around the world are targeted towards scientists and academics. Do you think that also there should be some culture or an effort, let's say, coming from the industry towards research?
So yes, I think that's an essential aspect. But again, in my experience, that's not lacking. So industry is actually very interested to be able to engage with research and to be able to engage early to find out what developments are going on. And I think it is, you know, an example is the Open Targets initiative that I described. Obviously, pharmaceutical companies have a problem that they no longer can afford to do all the research they would like to do, and they have to interact with academic organizations to do that.
Right, we are right on time, and I thank you very much, Professor Ian Matai. I thank all the person here who listened and intervened with their question, and I leave the floor for the next keynote. Thank you very much, Professor.
Thank you, Davide, and thanks to all the listeners. Thank you very much.
Have a nice day. Bye.
Thanks.